I'll get started. I think the first thing we're going to start with is a disclosure and acknowledgement. I'm going to be talking a lot today, obviously, about aptamers, and as you'll soon learn, aptamers have a lot of intellectual property associated with them, and a lot of the work that I'm doing, I do in concert, in a collaboration with a company, and the research that I'm doing has been sponsored to some degree by that company, which is called SomaLogic. Next, I'd like to acknowledge the people both at SomaLogic who've helped, and as well as people here at UW who have helped do a lot of the work that I'm going to show you, including the research scientist in my lab, Derek Thurstrup, Wei, a resident, and Lindy, who's been very helpful in the pathology department. So here's the things that I'm going to cover. What I'm going to do is not just talk about what we're doing in my laboratory and doing at SomaLogic, but I wanted to give you a flavor of what it is that aptamers could do for you. So we'll first of all talk about an oligonucleotide aptamer. I've given talks similar to this before and asked people, taking a show of hands, who's heard of aptamers? And some people put up their hands. And then I say, well, who's heard of aptamers but not from me? And they all go down. And so you'd like to just start with some definitions, talk to you about how an aptamer is made. The big sticking point about aptamers is, well, they're alternatives to antibodies, but what's the differences? And so we're going to talk about that. And then I'm going to talk to you about a laundry list of different things that people out there are doing, both out there in the world and here at UW and at SomaLogic with aptamers. So nothing is complete without a discussion of history. So this is where aptamers came from. 1990 here, published in Science. These two names are quite well known in the aptamer field, Craig Turk and Larry Gold, two folks at the University of Colorado in Boulder. And they were studying, of all things, a fairly non-biotech interested or clinical laboratory interesting subject. They were studying T4 DNA polymerase. So that's a phage. This is very, very old molecular biology type stuff. And they noticed that the T4 DNA polymerase, as all proteins that we know of are made, is it's coded for by DNA, it turns into RNA, and then the RNA is translated into protein. But what the T4 DNA polymerase does is the protein itself comes back and binds to its own mRNA at a loop structure, shown here, and it inhibits its own translation. So it's sort of like a feedback mechanism. We know about those things. Those things happen in bacteriology all the time. And so what Turk and Gold asked was, well, what is it about this sequence here, in this sort of stem loop structure, that makes it specific for sticking to T4 DNA polymerase? Does T4 DNA polymerase like to stick to anything? Or is there something about this actual loop here that makes it like to stick to that particular protein? So what they did is they synthesized a bunch of different versions of this mRNA, and what they did is they included a randomized piece here. So instead of including the normal sequence, which was AAU, AACUC, they included mixtures of all four bases. So 8 possible sites where you've mutated all the bases. That's 65,000 possible sequences. They took all those RNAs in a big mixture, and they applied them to T4 DNA polymerase, and they said, what sticks? And they washed off the stuff that didn't stick, and they took the stuff that did stick, and they took that RNA, they reverse transcribed it, amplified it by PCR, and now they took that family of sequences that sort of like to stick, and they stuck it again onto T4 DNA polymerase. And this time they did it a little bit more stringently, a little less time, a little more temperature, a little more salt, etc. And they did that iteratively over and over again to eventually evolve what is the best sequence of RNA that likes to stick to T4 DNA polymerase. And what they found, interestingly enough, after doing this, starting with a random library, is that they generated this sequence back, plus a couple other related sequences. And what they realized is, well, over the years and years, it looks like evolution has led T4 DNA polymerase to co-evolve with this structure to find the best possible sequence that it likes to stick to. But this process of making random oligonucleotides and seeing what sticks to a protein and partitioning them into ones that stick and ones that don't and continuing to amplify, etc., is probably a way that you could make RNA molecules or DNA molecules that stuck to anything. And so a sentence there they put, by extension, any partitioning agent may be used to educate RNA molecules. So this is what they came up with. So see this date here, August 3rd, 1990. This is one of the more unfortunate things in science. We have down here the 30th of August, 1990. So these guys got beat by 4 weeks. Right across campus, another group, Ellington and Stozak, were doing very similar work. And what they did is they made a big library of a bunch of different RNA molecules, and they all folded up into their own special shapes like RNAs do. And they selected now for RNAs that happen to bind to this small molecule dye called reactive blue 4. And so they found the RNAs that stuck to that dye. They washed away the ones that didn't stick. They took the ones that did stick. They amplified them up again. And then they did it over and over and over again, each time making it more stringent and eventually evolving themselves an RNA that looks like this. This one actually looks much more cool than the other one. It has several lobes on it, lots of stems and loops. And this thing sticks really well to reactive blue 4. And what they came up with was roughly 1 in 10 to the 10th random sequence RNA molecules folds in such a way as to create a specific binding site for small ligands. So the results of these two experiments were that people began to think, well, hey, if I want to make a piece of DNA or RNA that sticks to something, I should do something like what they did, and then I have a binding reagent. So we're left with now, I can't remember which group it was that named it Aptamers. I think it was actually the Gold group, but I'm not sure, which actually came up with the name Aptamer. But an Aptamer for these purposes is a short DNA or an RNA molecule that binds to its target molecule. What do I mean by short? I mean 15 to 75 bases. There aren't many aptamers that are less than 15 bases, and any more than 75 bases, it's difficult to work with these things. So that's a molecular weight between 5 to 50 kilodaltons, depending on what you do, what other modifications you make to the DNA. So small molecules. Now I want to make sure you understand this though. An aptamer does not bind to its target by hybridizing. It's not like an oligo you use for PCR, that the A, T, and G sticks to a T, A, and C. It doesn't have normal Watson-Crick base pairing. It might do that in itself. To itself, it might make a stem and a loop, but what an aptamer is doing is it's forming a three-dimensional structure that puts its chemical groups, like the bases or the phosphates or the sugars, out into space, and just like the variable region of an antibody, it sticks to something that's a three-dimensional shape, and it does it with some selectivity. So, what do we know now, 20 years after that we didn't know then? Well, originally, this was all done with RNA because the two guys who started were RNA chemists. Szostak actually just recently won the Nobel Prize for unrelated work, seminal work on RNA. And Larry Gold had always been interested in RNA stuff too, so that's why they started with this. But it turns out single-stranded DNA works just as well as single-stranded RNA for making these things, and that's what most people do. Very few people actually make RNA aptamers because RNA is not that stable. We all know that. Why does our body keep all of our genomes in DNA and not in RNA? It's because the RNA is not stable. There are aptamers now to literally thousands of different targets. Ellington's lab in Texas keeps a database of them. It's available at this URL. And there are thousands more, actually, that are owned by companies. There's a big difference between the aptamer field and the monoclonal antibody field, which I'll get to in a little bit, time-wise, but suffice it to say that most of this technology, up until very recently, has actually been entirely owned by someone. So one of the reasons you might not have seen a lot of this or heard a lot of it before is because it's being done inside of companies. And you can't just go and start selling aptamers because these companies here, SomaLogic, this diagnostic the company I work with, owns most of the rights under the patents to making diagnostics out of aptamers, and a company in Massachusetts, I believe, called Archemics has most of the patent rights for making therapeutic aptamers. However, the key patents are actually expiring. Remember I said this happened in 1990, so that's 20 years ago. Patents last for about 20 years. So it's pretty much going to start being open season on aptamer companies pretty soon. So how do you do it? Just briefly to remind you, if you want to make a DNA aptamer. So start off with a library. This is basically what they look like. So it'll have some known sequence of DNA, ATGC, something like that. A random sequence, so lots of ends. This is what you would see, what you would synthesize on an oligonucleotide synthesizer. And then another end here. So these sites here, you could have a PCR primer bind to later amplify it, okay? We have that random library here. And let's say we just know that the ones that I've colored there blue are the ones that are going to stick a little bit better to the protein we're interested in. Well, the first thing we do, going from the top right to the bottom right there, is we partition those DNAs, those single-stranded DNAs, into those that stick to something. So see, some of them stick to those protein molecules and some of them don't. These ones stick. We wash away the ones that don't, and now we elute off the ones that did stick, and now we have a more narrow, focused library of single-stranded DNAs that like to bind to that target. Now we amplify by PCR, and then we take this whole gamish and we put it back on. We isolate single-stranded DNA from it, and we put it back on the protein, but now we only let it sit for a little less time, or we add some more salt, or we heat it a little bit. And we do it more and more stringently until maybe usually 6 to 10 times you do this cycle, and eventually what you've done is you have evolved a narrow focused library of DNAs that like to stick to your target. At the very end of this, I might add, what's been traditionally done is you simply just take these pieces here at the very end of your thing and you just clone them into bacteria and then you sequence and you find out what was the sequence that worked. And once you found out the sequence that worked, you can then go back and just order it, synthesize it on a normal oligonucleotide synthesizer. However, there's been very interesting work shown recently which has shown that at this step here, after you go from here to this first step, you can actually take this enormous mixture and do high throughput sequencing on it at each round, and you can find species amongst the billions and billions of sequences that you detect that are getting more and less frequent. And you can get a good idea of what an aptamer sequence might be by doing high throughput sequencing. That's only been shown as a proof of principle. Right now, people mostly do the cloning. Well, I think, looking back way to the 1950s, I think it was maybe the work of Joshua Lederberg would suggest that those are actually the same thing. That a lot of things, like when you are looking at phage and doing some selection, or when you're finding a bacteria that has some resistance, a lot of the times what you are doing is you're simply isolating the one that was already there. So you're probably just selecting the one that was already there, and you're amplifying it relatively to its neighbors that don't like to stick. You can do this process by using PCR that is error prone to introduce mutations and such, but that's not normally what's done. So, caveats. I want to be objective here, and I want to tell you the things that aptamers can do and can't do. So what's the big problem? What do we know about DNA chemically? We know that DNA is a polyanion. I brought this in here, this picture here, to show you that all those little yellow and red things, those are all phosphates on the edges. So each of those little red things, that's a negative charge. So if a DNA piece is 50 bases long, it has 50 negative charges. That's a lot of negative charges. That can work for you or it can work against you. Let's say your target that you want to bind has 50 negative charges. Do you think the 50 negative charges on the aptamer are a good idea? No. If there's 50 positive charges on your target, however, that's great news, because the aptamer will now actually stick incredibly strongly and will kinetically probably seek out that target faster than diffusion because there's an electrostatic attraction. But this is why we have to be careful. So something the company I work with has figured out is that when you're doing that selection process, if you don't do anything about it, if you just do the same normal selection process I just showed you, which is called SELEX, S-E-L-E-X, so when you do the SELEX process, if you don't do anything, what you end up getting is DNAs that like to stick to positively charged things. And if your protein that you're interested in isn't positively charged, you don't really get anything. However, if you do the SELEX in the presence of a polyanion, something like dextran sulfate or a heparin or random RNA or DNA, something like that, that is sort of a decoy polyanion, that can actually occupy all those negative sites, and what you can start doing is selecting for aptamers that bind to things that aren't negative sites. So that's a really important thing, and we've been using that technology to help us a lot. All of the aptamers that we use in the laboratory are used paired with dextran sulfate. They are selected in the presence of dextran sulfate, and when we use them in the presence of dextran sulfate. And then the final issue, which people oftentimes talk about, is, well, how could you ever hope to make a DNA library, a DNA reagent that is possibly as diverse as an antibody? I mean, antibodies have 20 possible amino acids, but DNA only has 4 possible bases. So that's an enormous amount less diversity. Well, I don't need to do the math for you, but suffice it to say that a 20-mer randomized, or sorry, take it back, a 40-mer randomized library of DNA, if you had one piece of DNA, just one single piece of DNA that was 40 bases long of every possible sequence, the mass of that library would approach the mass of the universe. So it's extremely diverse, even having, just having micrograms quantities of random 20-mers. You have an enormous amount of sequence diversity and a lot of different shapes and sizes that can get then. But you're still limited. You really only have A, T, G, and C. And so what the company I've been working with does is they have started including, instead of using DT, so ATG, they don't use T. What they use is derivatized DUs. So what this is a triphosphate. This is deoxyribose triphosphate. This is a DU, and what they've done is they've attached a benzyl group onto it. And they use that in the DNA instead of using DT. So now, instead of DT, this molecule has benzyl groups, so big hydrophobic aromatic groups on it. And what that allows them to do is to introduce new functionality into aptamers, so make them more protein-like. The company has had success introducing benzyl groups into aptamers, introducing naphthyl groups and indole groups, so that like sort of tryptophan side chains. And also positively charged groups. They've also been able to introduce amino type groups on there too. So the nice thing about aptamers, though, is that it's really is just a problem of combinatorial chemistry. All the chemistry for this stuff has been worked out and is largely automated. So it's not such a big deal. So I think it's important now to move back and to put some context, because you're asking yourself, and the point I'm going to discuss in a little bit is, aptamers are pretty promising, and I think they're very cool, but where are they? Why aren't we using these things already? Well, I think a lot of it has to do with the timeline. If we look back at the timeline of antibodies, now, for some of us in the room, this is ancient history, and some of us, this was our professional careers, but 1960, the first immunoassay was published by Rosalind Yalow for insulin. It was 1975 that Kohler and Milstein published what we now refer to as monoclonal antibodies. 1978, Yalow won the Nobel Prize. 1984, Kohler and Milstein won the Nobel Prize. And then it was even in the 90s. It took even till the 90s, but it was still, that's still 20 years ago, that antibodies started getting used a lot for immunohistochemistry. So these are this is the time frame that clinical use of antibodies grew up. And for me to understand my bias against antibodies, I think you should understand where I come from. So just to give you an idea of what I was doing in 1984, that's me in 1984, that's me in 1978, that's me in 1975, and I wasn't even born in 1960. So here's the timeline for aptamers, though, much more relevant to me. As you can see, it started in '89 with aptamers invented. The SELEX process was patented in '90. Aptamers were being made in companies throughout the '90s. The SELEX process was automated, so that whole process of the PCRing and the selecting, that's all done on 96-well plates by robots now. That was done in 2000, and now here we are in 2010. And this company I work for has made aptamers to 1,000 different plasma proteins. So here I am, bright-eyed and bushy-tailed in 1990, ready to start to take on all of this, and now you can probably hardly recognize what's become of me after so long. There we are. It's beaten me down. So I told you, I promised you I would compare antibodies and aptamers. And this is important because the whole point of talking about aptamers is what can they do for us that antibodies can't already do. So antibody, you need an animal, or at least a cell line if you already used an animal. You need a rabbit, you need a mouse, you need a goat, you need a horse, something like that to make your antibody in. An aptamer, it's an instrument. You need a robot to do it. Robots can be complicated and break, and we know that they're difficult to work with, but scaling is a little bit easier. Antibodies are large. This can be an issue with diffusion if you want to use them to get into a small space inside of a cell or something. Antibodies are at the minimal about 150 kilodaltons for an IgG molecule, and as I said, aptamers can be 5-10 times smaller than that. Antibodies are proteins. I don't know how many of you people out there are protein biochemists, but proteins are hard to do biochemistry with. That's why there's a discipline, and that's why it's called a discipline, I think. DNA is actually really easy chemistry. As I said over and over again, it's automated. You can go on the internet and order yourself a piece of DNA, and it comes the next morning pure. You can't do that necessarily with any protein you want. Antibodies are relatively hard to derivatize. What do I mean by that? I don't mean that it's hard to stick a dye molecule onto a protein, but it's hard to stick 1 or 2 or 3 dye molecules exactly where you want to in a given protein. We can react with all the amino groups in a protein, but it's very hard to make a specific derivatization of a protein. Not so with DNA. We just type it in on the computer. If we have a phosphoramidite for that dye, we just type in dye, and it makes the DNA dye. Antibodies generally harder to purify than DNA. The time frame here is less of an issue, but vaccinating an animal, waiting, boosting, etc., versus that in vitro step, which takes a matter of days to weeks to do, SELEX, you actually get you can make more aptamers over a given time than you can make antibodies. But really, the whole problem comes after you've made them and figuring out if they work. And that time frame is pretty much the same for both things. But antibodies, one mouse, you get one antibody out of it. You immunize it. Whereas I didn't bring in a 96-well plate, but a 96-well plate's about this big, and you can make 96 aptamers in it. And if you have a stack of 96-well plates, you can make 1,000 aptamers when you're doing it. It's really just a question of how much you want to punish your robot. Antibodies tend to work in specific conditions. When you immunize a rabbit, the antibody it makes is going to react, it's going to bind to its target in whatever conditions the B cell or the plasma cell decides that it's going to work in, because whatever happened to be in that sick little mouse, that's going to be the conditions. An aptamer, if you make an aptamer and do that selection process in buffer A, the aptamer is going to work in buffer A. And if you make 1,000 different aptamers in buffer A, they're all going to work in buffer A. So that brings us to the last point. Antibodies don't generally like to work well with each other. The big rage these days is multiplex testing, doing lots of testing all at once. It's pretty challenging to find some salt condition, buffer condition that 75 antibodies will all work in, but not at all a problem for aptamers because they were all educated to work in that same conditions. So here are the current medical applications of aptamers. Here are the therapeutic ones. There's one FDA-approved drug called Macugen. It's an RNA aptamer to VEGF. It's heavily modified, though, to make it more stable. And I don't know who here has ever heard the saying, something's better than a sharp stick in the eye. This is actually not better than a sharp stick in the eye because it is actually a sharp stick in the eye. When you want to introduce this drug, you introduce it intravitreally. But it does work. It's just a particularly unpleasant way to take a drug. Other aptamers are in phase 2, 1, and 2 trials. As you can see, there's some aptamers to coag proteins, von Willebrand's factor, factor 9A, meant to treat hypercoagulability, and some other ones out there. And here for the final drum roll, here are the diagnostic applications of aptamers that have been approved by the FDA. There are none. So where are they all? Why not? Well, what I'm going to tell you about right now, and I wrote an editorial on this just last month in AJCP, and you could read about it for a little bit more detail, is what I'm going to call the thrombin problem. So this is actually a 3D structure of thrombin, and this is in complex with an aptamer that was grown to it. This is called the thrombin aptamer, and this is the sequence of the thrombin aptamer. It's a symmetric G-rich stretch of DNA, and that thing curls up into this structure, basically. It forms a pretty interesting structure. I bet you always thought that G bound to C, right? G triple bonds to hydrogen bonds to C, and that's how DNA pairs. Not so. What the G's do here is they form something called a G quartet, where 4 of them form in a planar situation, and it makes a structure like that, and it likes to stick to thrombin. If you do PubMed for aptamer and thrombin, you find hundreds of hits, and then there's even more publications on the thrombin aptamer if you search a little bit more deeply. And if I did this just before preparing this talk, and if I wanted to make this aptamer and I wanted a milligram of it pure, so a milligram of this binding reagent pure from an online oligonucleotide vendor, it cost me $29.95 plus shipping. Okay? So let's do aptamer science. Okay? So here's one option. Let's do aptamer science. Let's perform SELEX, so let's burn a postdoc for 6 to 12 months or spend a lot of money and get an automated platform to do it. Let's gather some candidate reagents. Let's screen them all. Let's develop and validate a clinical assay. Let's try and obtain a license from the patent holders, obtain clearance from the FDA, and then go to market with our great new clinical aptamer test. Or let's buy the thrombin aptamer for $30, put it on a silicon chip, write a paper, and do it again. What do you think people are doing? If you guessed option B, you're correct. Aptamers are to a large degree victims of their own success. It's so easy to work with them, and it's so easy to make one and just repeat what's been done or do a fancy new modification of detecting thrombin, that the analytical chemistry community has spent a lot of time working on a sexier way to detect something without thinking about that it's not so interesting actually to detect thrombin anymore. Some other aptamers that are like that are aptamers to ATP or PDGF. Those are some aptamers where you look at most of the literature out there has been published just on those ones. Even though there are thousands of aptamers out there, it's cheapest to just spend the $30 and work with a thrombin one. So, but why is this a problem? Why am I here telling you about all this? Well, why might they be better? I think that we can make reagents a little bit quicker. I think we can make them hundreds at a time, and this has been borne out by the industrial experience. I think we can store the reagents better. I don't know who here is interested in applications for the clinical laboratory in the developing world, but protein reagents tend to rot if they get warm, and they sit out for a long time in the humidity. DNA, the thrombin aptamer, you can boil it in 0.1 normal sodium hydroxide. It'll be fine. You can leave it out essentially on the bench top forever, and it will never go bad. If you're thinking about a clinical lab test where someone has anti-reagent antibodies, like human anti-mouse antibodies, well, how often do you think people are going to have antibodies to an 18-mer G-quartet aptamer? Probably not. Aptamers also have built-in sequence tags. So once you do some sort of a binding reaction, the way you measure it, you can use the fact that they are already barcoded. So you can turn the problem of protein measurement into a DNA measurement problem, and that's actually been solved already. People know how to measure the concentrations of DNAs and RNAs quite well in the array community. The kinetics can be better. They're smaller molecules. They can diffuse faster. And finally, the detection chemistry can be more varied. So I'm now going to give you the laundry list of the things out there that people are doing and the things that we've been doing to show you that aptamers can pretty much do all the things that antibodies can do, and I think a little bit more. So we'll start off with immunoassays. This is just to show you that aptamers can do pretty much just what an immunoassay can do. This is an aptamer to IgE. It was conjugated to a fluorophore in solution. That fluorophore and aptamer combination tumble freely, and when you excite it with polarized light, it loses polarization on the fluorescence emission. However, when that aptamer binds to IgE, it increases its fluorescence anisotropy. We have clinical assays that work just on this principle, very similar to this principle. And the point of this is to show you can make a fluorescence anisotropy immunoassay with an aptamer just like you can with an antibody. You can also do it for small molecules. This is an aptamer, a schematic of an aptamer that was made to L-tyrosinamide. And this is a floppy aptamer where the fluorescent label on it flops all around and loses its fluorescence if it's excited. However, when it binds to L-tyrosinamide, now the aptamer is now structured and rigid. The fluorophore is less floppy, less tumbling, and the fluorescence anisotropy increases here. Something I'll show you of some interest here. Look here. The KD for this is about, let's say, 50 nanomolar for IgE, and the KD for this one, hard to see, it's about 5 micromolar for L-tyrosinamide. This is a weakness of aptamers. Aptamers have pretty good affinities. Usually, the ones that I work with, sub-nanomolar, but low nanomolar are better for proteins, but really only mid-micromolar KDs or sensitivities for small molecules. Aptamers are not as good at binding to small molecules as antibodies are. So that's an important thing to think about. You might think to yourself, well, who cares about L-tyrosinamide? This is another one of these stupid things. Who needs to detect this? There's a really great aptamer out there to cocaine, and you can do the same sort of thing for that. So we could do a clinical assay for cocaine using a cocaine aptamer, if we so decided. And that's a molecule that people care about clinically. What's something that DNA can do that protein isn't good at doing? It can conduct electricity. DNA is like a little wire. It's actually like a semiconductor wire. When it's stacked in a big double helix, down the double helix, electrons can go from guanine to guanine to guanine to guanine to guanine down, and if you put it on a gold surface, you can actually detect current flowing through a piece of DNA. So let's make an aptamer here that has let's make a stretch of DNA that has a kink in it here, so no current can really flow in it, unfortunately. And in this kink, we put an aptamer that binds to something, in this case, adenosine, okay? When the analyte binds, it straightens out that kink. The DNA is now linear, it conducts electricity, and we can now detect current. This has been done to detect adenosine, and it can detect it at a very low level. So we can detect electrochemically in ways that we can't detect using proteins and antibodies. Another type of thing that you can do with DNA, with aptamers, is treat them in a different way. Don't treat them simply as binding ligands, but ask yourself, are there cases where the DNA itself can actually be catalytic, like an enzyme, like a protein enzyme? Proteins can assume certain shapes and do chemistry. Why can't aptamers? It turns out they can. There are 3 general types of DNazymes. This is a portmanteau word, obviously DNA plus enzyme. So there are some types of DNAs or RNAs that can fold up and do some sorts of chemistry. We are full of RNazymes in our bodies. That's what the ribosome is. The ribosome actually, that makes all of our proteins, is an RNA enzyme that's catalyzing those sorts of things. So some types of DNazymes or RNazymes, they catalyze DNA or RNA breakage or ligation. Some of them, the DNA can just curl up and do some sort of a reaction. You can make a DNA aptamer to the transition state analog of some chemical reaction, and then that DNA aptamer will catalyze that reaction. And then finally, sometimes the DNA enzyme acts simply by just binding and stabilizing a cofactor. And then the cofactor does most of the work. And that's the one I'm going to talk to you about. Quite some time ago, looks like about 12 years ago here, a group up in Vancouver raised an antibody to this porphyrin. This is N-methylmesoporphyrin. Not terribly interesting until they found out that this aptamer, which is the sequence given here, can actually bind to hemin. We care about hemin. That's an oxidized version of the heme group that's in all of our hemoglobin. What does it do when it does that? Well, it turns out that hemin itself can peroxidize things. So it can, if you add hydrogen peroxide on some substrate, it can alter that substrate and make it turn colors. It's really pretty bad at it. This is a plot of the reaction rate versus the concentration of hydrogen peroxide. You can see hemin alone is actually pretty poor. But when you add the DNA and now make the holo DNA enzyme, the DNA plus the hemin, it actually really catalyzes the reaction quite well. So what this is a piece of DNA that acts like the enzyme peroxidase. Okay? Why is that interesting? Well, let's say this gets a little bit confusing here, but I'll just tell you that that DNA enzyme, it only really likes to work in the presence of potassium, just the way it works. Okay? Lead, it hates. It does not work in the presence of lead. So what can you do? You can actually make an assay for lead now. If you have some sort of assay where you can use this DNA enzyme in the presence of potassium to catalyze some reaction, to say make luminol turn into a luminescent reaction product, in the presence of lead, it's going to disrupt that DNA enzyme formation, kick out the hemin, and now it's not going to work anymore. And what we have here is a plot of lead concentration, log lead concentration, so down here to nanomolar up to micromolar lead, and here's the chemiluminescence. And as you can see, lead inhibits the chemiluminescence of this DNA enzyme. So what we have now is a way to detect lead concentrations. What we asked in our laboratory was, I don't really care so much about detecting lead. We can do it by mass spec a lot better. But I'm interested, when I saw peroxidase, I thought, well, one place in pathology where we use peroxidase is immunohistochemistry. Can I start thinking about replacing antibodies in immunohistochemistry? Let's start by seeing if we can remove the peroxidase part of immunohistochemistry that is usually used to develop, to make the brown stain that you see when you see immunohistochemistry. So this is the molecule that's used to make the brown stain that we all see in immunohistochemistry. It's called 3,3-diaminobenzidine. And the first question I asked was, does that DNA enzyme oxidize that molecule into something brown? And on the left here is agarose beads that I labeled with a control piece of DNA, not the DNA enzyme. And these beads have the DNA enzyme on them. And a few minutes later, after adding a DAB, this molecule, and hydrogen peroxide, it turns them brown. So can we do immunohistochemistry? And in fact, yes, we can. This is a piece of prostate tissue stained with a PSA antibody. This one had horseradish peroxidase added to it. This one had the DNA enzyme added to it. In fact, we can turn it brown. So this is still actually working quite slowly. It takes a lot longer. It's not nearly as fast of a reaction as horseradish peroxidase. Horseradish peroxidase is probably one of the best evolved enzymes on the planet. It works almost at the diffusion limit, and the DNA enzyme still works maybe 1,000 times slower. But sort of proof of feasibility that if I wanted to start doing immunohistochemistry with just DNA and DNA technology, can we do that? Well, I think maybe. So, next thing I'm going to talk to you about is getting on to these contentious statements I made where I've told you that the aptamers all like to work in the same conditions and we can do multiplex assays with them. So, what do we have? What is the problem with antibodies? As I said, it's hard to make antibodies work 20 or 30 or 40 or 50 together, all the same conditions. It's practically difficult to do that because antibodies don't have any sequence tags on them. If you're going to make 20 of them all together, you have to stick them to 20 different things that you can then separate. The best technology for doing this right now is a Luminex platform, which they have different beads, and their best technology has 500 different color beads, so they claim to be able to separate maybe 500 different antibodies. But practically, there's no real Luminex kit that's good at measuring more than 20 or 30 proteins all at the same time. So antibodies are okay for targeted assays, one or two things at the same time, but not great for measuring lots of things at the same time to do discovery. So how might you make a proteomic array to measure the concentrations of lots of things all at the same time using aptamers? This is what the company I work with, SomaLogic, does. They mix 800 and some different aptamers with proteins in serum, and they get a bunch of complexes. Some of them are the correct complexes here, and some of them are incorrect complexes. These little squiggly lines are the aptamers, and just to be clear, this little linker B, this is a biotin which can be cleaved off by shining a bright light on it. It's a photocleavable biotin, okay? So they make all these complexes, they stick them to a streptavidin bead, so now all these complexes are stuck to the beads. They wash off everything that doesn't stick. They now come down here, and they add a reagent that biotinylates all the proteins. So now all the proteins have biotin stuck on them. They then add that dextran sulfate molecule I was telling you about that gets rid of all the nonspecific interactions. That kicks off the nonspecific interactions. They then photocleave, and now what they have is these complexes in solution now, which then they bring down to another streptavidin bead and through the biotins on the proteins. What they're left with at the end here is you elute off the aptamers. And this is a pretty complicated reaction scheme, but what I just showed you, what it actually does is it converts the DNA concentrations that you started off with, the protein concentrations you started off with into DNA concentrations at the end. They have to do all these steps because they don't bother making sandwich reagents. They don't have 2 aptamers to everything, they have 1 aptamer to everything. So if you want to do a conversion of protein concentration into DNA concentration, this is one way that it works pretty well. At the very end of this process, you take these aptamers, you plate them down onto an adjuvant array, and you just sort of see how bright is the fluorescent spot that I see, how much DNA was there. So how does it work? Well, they have 825 of these aptamers, and they all work well. They can measure quite quantitatively 800 things all at once. Mass spec discovery doing 800 assays, difficult to do with 15 microliters of sample. They can do this assay on 15 microliters of sample. In discovery mode, as I said, it's read on that adjuvant array. In a targeted mode, though, you can do one of these things, just use one of these things as the reagent, and you can read it out on a Luminex, you could read it out on, you could do quantitative PCR, just to PCR out the sequence, etc. But the big power is that the discovery assay that I just showed you is the targeted assay. So after you do your discovery thing, where you look at 800 things all at once to try and figure out what are the interesting biomarkers, you then just select the 3 that worked, and then you just use those 3. It's the same assay. You don't have to go through this laborious process of converting mass spectrometry findings into an immunoassay. And I might add just again that this whole thing is automated and done in 96-well plates. So does it work? That's more important. Here's some of the data. This was published by SomaLogic. I had nothing to do with this study, where they took a bunch of patients with chronic renal disease and they measured 800 some proteins in them, and what did they find? Well, here's one we know about. Here's cystatin C. As you can see, as the creatinine clearance, sorry, that's a little small. The creatinine clearance goes down, cystatin C goes up. Well, we all knew that, right? So this is just to show you that it works. What we might not have known, however, that the TNF soluble receptor 1 does the same thing, as does matrix metalloproteinase 7, as does pleiotrophine, as does factor D, etc. So they discovered a lot of new markers for chronic kidney disease. Chronic kidney disease, I mean, we can already do creatinine. That's not such a hard problem, but do we have other things that this technique can be looked at? This is once again another thing that I didn't participate in, but they took a bunch of samples, thousands of samples from patients who did or didn't have lung cancer, and they did this discovery platform and trying to find markers, things that were quantitatively increased or decreased in patients who had lung cancer. And they found some interesting hits. They found that cadherin and I don't even know what all these things are. Stem cell factor soluble receptor went down, and other things like CD30 ligand and endostatin went up in lung cancer. You combine all these things into a multivariate marker, a multivariate classifier. In this case, they use a naive Bayes classifier. And this is an ROC curve. We're used to looking at these in laboratory medicine, and you can see that this sort of thing performs well. I wanted to show you this data, though, to really show you this thing on the bottom. What is it? On the bottom here is a little hash mark, is each individual marker that they looked at, so all 800 of them. The top line here, if it's yellow, it means it's a bad marker, and if it's blue, it means it's a good marker. And the top line here is it a good marker for detecting lung cancer? And the bottom line is it a good marker for detecting where your blood got drawn in the study? So pre-analytical variation. And what you can see is they got a bunch of markers that are really good at telling you whether or not you have lung cancer, and they have another bunch of good markers that are really good at telling where your blood was drawn. And they actually have some markers that seem to be pretty good at telling you whether or not you have lung cancer. And horribly vary between the sites, so you wouldn't want to include these things. But this is the power of doing huge discovery with hundreds and hundreds of things. You can start asking these questions. Not only is something a good marker for disease, but is it going to be a good analyte later down the road? So what is my collaboration with these folks? Two things. The first thing is on a proteomic study, largely to get at the question of neurodegenerative diseases. Neurodegenerative diseases are hard to diagnose. Sometimes you need an autopsy. I'm not sure anyone here would want to undergo that procedure to diagnose their disease. So people tend to look at CSF because blood tests are hard to, have not been terribly successful. So we've been looking at the CSF proteome in normal patients and patients with neurodegenerative diseases, trying to ask ourselves, first, can we use this technology to measure proteins in CSF? And second, can the results predict disease or tell us something interesting about the proteome with aging? And the answer needs math. And I'm real sorry about this. I'm going to do this in just a few seconds. But everyone in this room can think in 3 dimensions, right? This is three dimensions of data here that I plotted here. We can all think in 3 dimensions. You can drive a car, you can think in 3 dimensions. If you can back the car up, you can think in 4 dimensions. But what about 825 dimensions? Can anyone here think in 825 dimensions? I cannot. So is there a way that we can think about 825 dimensions in less dimensions? And the answer is yes. So let's say we measure 3 gene or 3 protein concentrations, and we get this cluster of different, these different data points here of several different groups. We can't make sense of it. I mean, I could put on a glove and try and move it around in virtual reality space. Or what I can do is I can put a new 2-dimensional plane inside that 3-dimensional space and sort of figure out what is the projection of that data onto this 2-dimensional space. And now I'm left with this. If I move all of these data points now onto 2 dimensions, what I see is this. Hey, that looks great. Now I can actually start thinking about those 3 dimensions, but in 2 dimensions. It's easier to think about it. Furthermore, if I want to make it even simpler, I can reflect all of this data now onto the x-axis and now think about it just in 1 dimension. So what I just did is I went from 3 dimensions of all that data down to 1 dimension. This process is called principal component analysis in general. This is a gross oversimplification, but what you need to know for today. And if the mantra of what principal component analysis does is what it tells us is how there are correlated patterns of variation in the data, okay? So this is data that we did. So we took 3 different groups and we measured their CSF proteins and we did that analysis. And here the blue guys seem to separate out from the red guys, seem to separate out from the green guys. But on this one, unfortunately, this is the unfortunate one, the red guys don't seem to separate out from the green guys. So we can't really tell the 2 groups that are different by this analysis. So what do you do when you're left with something like this? When you're left with this sort of analysis, you can do something called disease-specific genomic analysis. This is a technique that has become quite popular in the genomics and the array fields. And basically what you do is you do that sort of analysis you do principal component analysis on normals, and then you remove the variation that you see in normals to try and get at what's going on in the disease population. Because sometimes the variation you see in the normals is much, much, much more than you would normally see in a diseased person. So you have to ask yourself, is there some way of removing it? So if we do that to this technique here, we remove out all the variation in the normals. These green ones are control patients. Right now they overlap with patients with Alzheimer's disease, okay? But now let's remove out all the variation that we see in the green patients, in the green controls, and now we actually can see, look, the Alzheimer's disease patients are over here and the control patients are over here. So we're getting at, by looking at hundreds of different protein concentrations in the CSF, we can start thinking about, we have maybe markers that are going to tell us something about whether or not a patient has Alzheimer's disease. Here's another thing we looked at. This data I showed you was actually aging data. So these are young, these are middle-aged, and I got in trouble when I was writing the grant application on this because I referred to the 65 to 90-year-old group as elderly, and then I was told that, you know, some of the people that are reading this grant are going to be 65 and they're not going to be like getting called elderly. So we're going to have the young, the middle-aged, and the distinguished groups here. And as you can see, they're separated out. And then we define, like say, this has actually been known, activin A, this is the signal from the Aptamer we saw, and these are all about 120 different control patients. We see a great increase in activin A. And we have a lot of other markers that we found that are novel, that are interesting, potentially interesting, very interesting markers of age. Now, why would I care about just looking at age? These are all normal patients that we mentioned. Believe it or not, there are subjects who volunteer to go have lumbar punctures done for research. And all these people did it, hundreds of them. We actually have probably 900 more that we've yet to analyze. But what we can do is we can look at their CSF proteome and we can make up a classifier based on the concentration of those proteins, and we can predict how old they are from the concentrations of those proteins. So here is the predicted age, the patient's real age, and here is their predicted age. Now, why would I care about this? Why would I care about predicting your age? Well, there's a big question. A lot of people wonder, is there a difference between people's chronological age and their quote-unquote biological age? I think my wife would probably be pretty interested in this because I think she's fairly certain at sometimes that I have the brain of a 14-year-old. So I would probably come down somewhere around here. But I'm not going to volunteer for this study. But one thing we don't know is maybe our patients with Alzheimer's disease, they get it? Do they have old brains? Is there something, when people are cognitively impaired, do they have older or younger brains? We don't know. This is a tool that may be able to start answering those sorts of things. So I'm going to finish in the last couple minutes with two further examples of things we've been doing in my laboratory. So far I've talked really just about clinical laboratory types of things, quantitative fluid-based assays. But do aptamers have any place in tissue? And the answer is yes, they do. With several limitations, it has been possible to replace antibodies as the binding reagents in immunohistochemistry. This is a breast carcinoma that overexpresses the HER2 protein, probed with a primary rabbit monoclonal antibody, and done immunohistochemistry. Routine immunohistochemistry is done. And over on this side here, this is a similar overexpressing breast carcinoma that expresses the HER2 protein. A HER2 aptamer was stuck to it. It was developed in the same way with avidin biotin peroxidase, and this was done. Now, this is done on frozen tissue, on the right here, frozen tissue over here, and fixed tissue over here. The aptamers I've been working with were raised to native proteins, so they tend to bind to the native structures of proteins. Fixed tissue, not native proteins anymore. And so none of the aptamers that we're working with yet have been able to bind to things in fixed tissue. So our next step for this work is going to be to raise aptamers to formalin fixed proteins, and hopefully they'll work for immunohistochemistry in that way. I just have to blaze through this. It's often very depressing when you're at a talk and you see like someone's thesis got shot up in just one slide and they went through it. And that was all that work. So this has been an enormous amount of work of the next two slides I'm going to show you, but I got to go through them quickly. But this is aptamer histochemistry of EGFR on the left there, the epidermal growth factor receptor in human epidermis, and on the right you can see an immunohistochemistry shot of a similar thing. You can see that it binds to the same thing. This is just done by fluorescence because it was easier to do. And then next I also have a picture. This is PSA. Aptamer histochemistry. This is a frozen section of prostate, normal prostate, and as you can see here, the glands, the cytoplasm of the glands are stained by the aptamer. This is actually an aptamer conjugated to a semiconductor quantum dot, and this is something that's a little bit more difficult to do with antibodies, but something that's not actually terribly difficult to do with aptamers. So, last thing, can we discover tissue biomarkers? So we can measure hundreds of things in CSF or in plasma and discover biomarkers, but sometimes you might as well just go to where the money is, actually, and look for biomarkers in tissue. So Wei helped out in this, and we collected a bunch of lung cancers and adjacent normal tissue, and we ground them up, and we looked in the lysates of those tissues. Can we find biomarkers that differentiate between the patient's lung cancer and their adjacent normal tissue? And the answer is yes. We find some biomarkers that are elevated in the tumor and some that are quite low in the tumor. And then the other nice thing that we're able to do is, for this biomarker, we're able to find that the biomarker's concentration is elevated in the ground up tissue. And then we came at it and we did immunohistochemistry, or aptamer histochemistry, with the same reagent that was used in the discovery platform here. We then used here as sort of a diagnostic reagent. We found that this biomarker is highly expressed in the stromal matrix of tumors, not expressed in the tumor cell nests, which are the tumor cell nests, which are right here, but expressed in the stroma. So that was a lot. It was a big survey. What do I want you to take home from this? What I want you to take home is that aptamers have been around for actually a very long time. And there have been a lot of limitations, a lot of reasons why we're not using them all right now. I went over them. But there's a lot of potential benefits over antibodies. There's a lot of potential applications. All the things that we can do with antibodies, we could do with aptamers too. And we could do even more, because at the very beginning, we can do the discovery work, and then the point I wanted to make is that the discovery reagents, the discovery process, is the same thing that we could eventually then use to do the diagnostic test at the very end. The problem, however, is I've told you about lots and lots of potential, and we need to turn this potential into actual. And so with that, I will leave you and say thanks, and I'll take questions.
People used Aptamers to introduce lethal molecules into nuclei of cells?
Yeah, so the question was, have people used Aptamers to introduce lethal molecules inside of cells? Into cells, yes. There are some Aptamers that are small enough that they penetrate cells okay. Some of them need to be carried by maybe one of those membrane, those lysine-rich membrane permeant peptide things. But I have seen papers where people have been able to introduce molecules, Aptamer molecules, because they were small enough that they will be able to get inside of a cell.
Aptamer molecule to be a carrier for, say, something that emits alpha radiation.
Yes, that sort of thing has been done. Susie.
Can you talk a little bit about stability and the stability of the thrombin Aptamer? When you introduce it into people, though, there's endogenous DNases. What is the stability?
So the question is, what is the stability of Aptamers in vivo? And that's a big problem. So in histochemistry, it's not such a big problem because you can kill things, you can fix things in ethanol or formalin or whatever. But in people, you're right. An RNA Aptamer's half-life in a human being is a few seconds. A DNA's Aptamer's half-time is a few minutes. The VEGF Aptamer macugen has been heavily, it's been pegylated, and it's had fluorines and methoxy groups put all along it. All sorts of different things that have been done to it to prevent nuclease destruction. All of the Aptamers that I work with all have an inverted DT stuck at their 3' end, which prevents 3' exonuclease activity, which is one of the more common things that happens. Mostly DNAs in vivo get chewed at from either end and not endonucleased. They don't get chewed at in the middle. So minutes, unless you do something to them, is the half-life. Mark.
Another push for immunoassays is to make them faster. Are the reaction rates for fluid-phase, serum-based immunoassay likely to be faster, slower, or is there a way to manipulate that so they would be very rapid?
So the only data I have on that, I can say, this isn't actually fluid-phased. I unfortunately don't have my best picture of it, but the HER2 Aptamer, you can apply. It is essentially completely bound to its target in a minute. Their on rates can be quite rapid, essentially diffusion limited. And as I said, in the case of electrostatic driving, they can actually be much faster than diffusion. But largely the reaction, like the proteomic array that is done, there's no turnaround time need. And so the way it's done, it's done over a day actually. And so there's no real reason to make it go faster. So it hasn't really been tried to make them to go really, really fast, but there is good reason to think that they would perform at or faster than antibodies. Again.
And then patients with autoimmune disease, lupus, certainly have anti-DNA antibodies, both single-stranded and double-stranded DNA. Do you think that's likely to be an interference with these as reagents in serum and plasma?
That's a very good question, and we'd actually we'd have to test it. I don't know that double-stranded or single-stranded. So what would be mostly interesting would be single-stranded DNA antibodies. I don't know that they would end up binding well to hairpin loop structures, things that are only 15 to 20 bases long as opposed to long stretches of renatured or double helical DNA. I don't know, honestly, but you make a good point, and so we can't know until we try. Morema.
That was a great presentation. So I have a follow-up question. Can Aptamer be immunogenic?
Can an Aptamer be immunogenic? They have not been to date, the times that they have been assayed.
So Jeff, how much longer do we have to wait, and what's your predictions as to what the first products that we are going to see?
So the lung cancer proteomic, the lung cancer proteomic test right now, the company that I'm working with, as I said, this has nothing to do with me. They are partnering with Quest Diagnostics and planning on offering this thing soon, I think within a calendar year, as a diagnostic for lung cancer. Whether or not, that's, I think that in a business perspective would count as a forward-looking statement. So I don't know. I mean, I don't know their business plans. But that certainly is their plan to do that. And they have other assays too. They're looking at ovarian cancer markers, et cetera. A lot of the big ticket ones that people have been trying to do for a long time. But their lung cancer data has been published now and in a peer-reviewed journal and has been That's, I think, what they're really hoping to go to market with first. And they're also thinking about just trying to start selling these reagents directly to people who are interested to use them. Anything else? Thank you. Oh, thanks. Okay.